Video 6. Laboratory Testing Techniques. In this video, we'll discuss the testing performed at the State Public Health Laboratory. What happens to samples once they are dropped at the lab? Specimen receiving. For water-related outbreaks, water samples are processed at the State Public Health Laboratory in Nashville or Knoxville. To reiterate, ensure the chain of custody form is complete, with the date and time that the samples arrive at the laboratory. Maintain a copy for your records. Laboratory staff enter data from this form into StarLIMS, the laboratory's testing database, which generates a barcode label. This barcode label is attached to sample containers and is key to tracking samples and test results. For Legionellosis outbreak investigations, the water samples and swabs are delivered to the clinical laboratory for Legionella testing by polymerase chain reactions or bacterial culture. First, the water is filtered. The resulting pellet is analyzed by PCR. Test results are entered into StarLIMS and emailed to the outbreak investigation team. This process may take up to 7 days. The environmental microbiology team conducts testing for total coliforms, E. coli, and conducts large volume filtering. Water samples, other than for Legionella, are initially processed in the environmental microbiology lab. During gastrointestinal outbreak investigations, samples will be collected for total coliform and E. coli quantification and molecular testing by PCR, including BioFire film array, which includes a screening for 23 gastrointestinal pathogens. For total coliform and E. coli concentrations, IDEXX ColiAlert fluorescence is used to estimate the most probable number, MPN, per 100 milliliters. After incubating the samples, laboratory staff review the tray for color change and fluorescence. These changes allow them to calculate the total coliform and E. coli concentrations. Large volume samples, over 10 liters, for BioFire or other PCR testing are first filtered in the environmental microbiology lab. The filters are then transferred to the clinical lab for more specific testing by PCR, whole genome sequencing, or culture. The PCR, culture, and whole genome sequencing tests conducted at the lab help to further identify pathogens involved in outbreaks. Laboratorians rely on us to correctly document, collect, and transport water samples for testing. It takes a team to successfully investigate waterborne outbreaks. Thanks for being a part of the team.